So we posted it, so let's say within 4 days, which was really nice. I really liked the response of the participants. And mostly the crowd is from masters. So I am sure that they would able to pick up, the imagining techniques and things like that. Perfect. We are live. Great. So thank you so much, everybody. So good morning, everyone. Welcome to this exciting online session of the SciComm Synapse. I am Dhairya Rajguru, curator admin, and I am delighted to have you all here. We are honored to host Dr. Abhijit Day today for an insightful talk titled Imagining Molecules at Work, Illuminating Disease Biology in Vivo. Dr. Day's research is at intersection of imagining and disease biology, revealing how molecules function within the living systems. This session promises to offer a fantastic look into how cutting-edge imaging techniques helps us better understand diseases at molecular level. Stay engaged. Please drop your questions in the YouTube live chat. And now I would like to request our teammate Bhakti Sadguru to please introduce Dr. Abhijit Day today for the event.
Good morning, everyone. It is my distinct honor to introduce Dr. Abhijit Dey, a leading figure in cancer research and molecular imaging. Dr. Dey completed his PhD in genetics at Banaras Hindu University in 1998. Following his doctoral studies, he pursued postdoctoral research at the for Cellular and Molecular Biology in Hyderabad and further honed his expertise at the Crump Institute of Molecular Imaging, University of California, Los Angeles. In 2009, Dr. Dey joined the Advanced Center for Treatment Research and Education in Cancer and the Tata Memorial Center in Navi Mumbai, where he currently serves as principal investigator. His research is at the of developing non-invasive imaging techniques, including micro-PET, micro-CT, and optical imaging to enhance cancer diagnosis and therapy. Dr. Dey's prolific contributions to the field are evidenced by over 80 publications and a US patent for bioluminescent resonance energy transfer technology. His work has significantly advanced our understanding of disease biology in vivo, bridging the gap between molecular insights and clinical applications. Today, we have the privilege of hearing Dr. Dey's lecture titled Imaging Molecules at Work, Illuminating Disease Biology in Vivo, where he will share his innovative approaches to visualizing and understanding disease processes within living systems. Please join me in welcoming Dr. Abhijit Dey. Thank you.
Thank you, Bhakti. Um, good. We are in the morning in India. I believe all of you are at different places in the world. So, good morning from my end to all of you, and thank you for joining. I was talking to the hosts just before it started, and now it already looked like I'm behind a curtain of pinkish curtain. But believe me, I've joined from my computer. Maybe some corruption in the camera setting is causing this. But I'm live, and I will take you through this exciting journey that I had the opportunity and privilege to see this world of research gaining a huge momentum through this technology development on medical imaging. And the imaging that I would be mostly talking about is a diagnostic body imaging. So let me first try to share my slide. Um, Believe me, I would be actually, uh, it should be entire screen. Share. So, can you all see?
Yes, sir. It's visible.
Okay. Can you see the transition? Fine.
Yes, sir.
Okay, good. So under this talk title, so I am at, as introduced, I am at Tata Hospital Research Center, which is called Advanced Center for uh, Training, Research and Education in Cancer. It's located in Mumbai, Navi Mumbai, actually. And I have been here serving as a scientific officer for the last 15, 16 years now. And so we are primarily researchers who are involved in the disease biology, meaning it means to us is the cancer. And my group is primarily focused on breast cancer. So taking some of these few of the examples from my past research, I would taking my intention is to take you through the journey of imaging and how the different developments has empowered us in visualizing molecules which work at the inside the cell. So if I feel free to interrupt me if you want at any point of time. I'll be speaking my mind, so it won't disrupt anything. So I would be happy to take on questions as I go. So let me just start with this comparison that human body itself is a opaque system, and what is really going on inside our body is always a matter of curiosity, and a greater understanding, and the deepest understanding possible can only help us address the disease, any disease per se. And we also know that the body system is actually there are a lot of different organ systems, and each organ has different types of cells. And then, so basically, cell is a unit. And then, on today's biology knowledge that we already gained, we know that human cell system itself has multiple subcellular components, and there are complex chains of reactions. And today is the omics era, right? So in this era, we get to know that so many genes, so many proteins, and their interactions, their actions, interactions. So all that matters. And uh, when everything goes fine, we go fine, meaning the normal function. And if something start getting corrupted, and that's where the disease onset starts, right? So, so with that brief note, so the matter that I want to impress you upon is something like we must see we there is a greater need of visualization, and when visualization comes together with a quantified ability, quantification ability, then it's the greatest strength that we can gain. And this is exactly what imaging delivered, and that's why it has formed such a big strength in the modern research. So as I was mentioning that, the inside the body, there are a lot of different organ systems. Let me just take this highlighter. Yeah. So there are different organ systems, there are skeletal systems, there are muscular systems, and there the work wise, the function wise, they are widely different, and they are anatomically also they are quite different. Now, when we talk about body imaging, what we see is important, and how we determine what we see. So, if you just take the simple concept of image, what is a good image versus a bad image, right? So, what it differentiates is these four parameters primarily: the resolution, and the resolution can be of the how well we are resolving what we are liking to evolve. And there could be spatial resolution, meaning in space, meaning if there are two dots, how well they are separated. If there are two eyelids, how well they are separated. um, or they are looking like one. So that is a resolving power in space. And then the temporal resolution, meaning oftentimes, we are dynamic system, and a cell itself is a dynamic system. And then, of course, the body. And any disease for say, so there is also a time kinetics that how the disease grow over a period of time. So there are temporal resolution, and then also there are spectral resolution. So something like jaundice, if you can take an example. So there the body takes a tinge of color of yellow, right? Or another disease that takes oxygen deprivation per se, it takes a color of bluish. So there are such spectral change, and can we resolve uh, uh, that uh, well, and so that we can actually sense better what is actually the change that has happened. And then there are specificity and sensitivity. So they are interlinked in a way. So the sensitivity is basically the tool's ability to capture when the signal is very low, for say, like in camera, simple camera system. If you compare, then sensitivity is actually when it is a very dim light, can we still capture the image. Similarly, for medical diagnostics, so if the event is very early, so the expected signal that change that you expect is very low, and can we determine that change at that stage? So that is the sensitivity. So higher the sensitivity is better, and it will actually give the the strength for the specificity matter, because specificity is a determination of the false negative, for say. So so how accurately we are determining that a disease has started or not started. And that comes definitely from the sensitivity as well as the resolution. And then there is the field of view. Field of view is what we want to see. So if my organ of inspection is only eye, then the field of view is very small. It's about a couple of inch, and that covers the eye area. But if I want to see the whole body or the whole head, so the scale that it's changing, the spatial resolution that you can imagine, right? So what we want to see, what we want to bring in within the camera's ability to capture. So that is the field of view. So these four parameters, how it plays out. So now when we talk about imaging molecules that function in the disease, so you have to imagine the scale and the change. So it's a huge thing. So something like astronomy, what they do is far away and far away stars and planets. So they try to image and trying to resolve, and we have excellent strength gain on that matter as well. So similar thing in an opaque body system, there are multiple complex organs, and then inside the organ there are tissues, and then tissues are composed of cells, and the cells are composed of nucleic acids, proteins, and so on. So, and the scale that it varies from foot long to centimeter to millimeter to micron to nanometer scale, or even Armstrong scale. And in today's time, in the modern biology, actually, we are inside the change in the gene, or a single nucleotide or a protein, and their mutations, how it is aggravating the situation in an organ scale or a body scale. That is what it matters. So while looking at a body or an organ, fetching the information from what is in the happening in the DNA or the protein is the greatest challenge that imaging world uh, has faced in the beginning, but now a lot of it has overcome, and a very important development that has happened. So the other way to look at, and before I go for that. The cancer, the disease that I'm mostly focused, as I mentioned. So, if we have to linearize this in a very simple term, because I believe a lot of you are in your early stage and have not gone deeper inside the cancer biology. So, it's a disease that caused by uh, you know uh, the leading cause of death in the human, and it's uncontrolled. So the cells that are, it always starts in one cell, and then that corrupted cell which get uncontrolled growth pattern, and then it accumulates its number within that organ, and until it pushes a boundary within that organ, and then the organ function starts signaling for difficulties. Only then we start realize, right? So it's a change in the cell function, and over a period of time it aggravates, and then there is a structural change that happens, which is what the first symptom when we showed up in when the disease is detected, and it is already a change in the structure. But the disease actually had started much earlier, when a single cell stage, which we had no clue. And sometimes it takes many, many years to realize that, because that one cell to multi-cell or a organ scale change, a couple of centimeter of cancer, is a sometimes many years it takes. And then at the latter stage, it has the propensity to spread across different organs. So the factor of time is a very important issue. And then the and when we talk about early detection or cure of cancer, so this is where it is important to know that many cancers today have found a solution, even though majority of the patient that shows up in our clinic is at the late stage. But many cancers can be, I mean, our position in terms of curing cancer would have been much better if we could have detected the cell function, early change in the cell function. And that is what imaging world is promised to provide in the future run. And that is a challenge, but we have progressed, and future world will be even more better. So this was just the cartoon I tried to show that a cancer came, and then at a later stage it has metastasized to different organs, and when it goes to reach out to a critical organ, then the disease aggravates. Other way, there are even deeper understanding. Again, I'm very simplifying the issue of resistance. So a lot of cancers that patient that they show up, they have a treatment, and they undertake that treatment with the hope that they will be cured. But then 5 years, 10 years later, again the disease relapse. So why that happens? So there are a couple of different hypotheses that stays there. So some says that the cancer itself is a heterogeneous disease, so there it is not composed of one type of cell. There are multiple types of cells, and when I am treating this disease with a particular drug or a set of drugs, then I'm selecting, I'm killing, I'm being able to nullify the some of the percentage of cells, but some percent remains, and that continue to grow, and then the disease. So this is called inherent drug resistant cancer cells. So they never responded, and they formed a colony, and then they bounced back. The other theory was that, it was a bit older theory that we all now believe that cancer is heterogeneous, but it was a homogeneous mass, if you consider. And then when you apply the theory, therapies, then what happens is that some of the cells, they manage to re-network uh, uh, and escape the therapy. So in some sense, they are like an intelligent body system, and some people can call them as smart cancer cell or cancer stem cell or so many other terms that is there. So, and these escapers, they can actually bounce back and give the recurrent tumor, right? So, and these are called acquired drug resistance. So, in the beginning run, the cancer was responding, but at a later stage, it is made up of a completely different set of cancer cells which do not respond to that therapy, and so the cancer bounces back. So, with this very basic concept in place, let me also tell you the couple of factors that are molecularly how cancer can be simply known. So, cancer is always gene related. So, I'm calling it not genetic. Some people call it genetic. It is also genetic. Genetic in the sense it can be hereditary, but there are also the gene involvement. Not necessarily all cancers are hereditary, but there are gene involvements always there. So the change that happens in a oncogene, either a mutation or a overexpression or suppression of tumor, the tumor suppressors, which actually these are the set of genes that are growth inhibitors. And if they are nullified, then if their presence is lost, then the aggravation of that control mechanism goes away. So either the oncogene or the tumor suppressor genes, and oftentimes the changes that happens is through the mutations, their copy number change, overexpression, or in case of tumor suppressor, their suppressions, and which cause the normal protein change in the normal protein function. And then the mutation may occur during the cell division by external agents. So either a viral carcinogen or chemical carcinogen, environmental carcinogens, so pollutants and so on. And and there are even inherited genes. So one of the great examples that many of you know is the BRCA, breast cancer related, and it is in the female. So the breast and ovarian cancer, the BRCA carriers, and there are many other inherited cancers. So and then the most cancers result due to mutations in the somatic cell, and rarely there are also germ cells related. So now, if we bring back that same concept that the dynamics that is from the change in the cell function, where all these different parameters, different functional aspects that got modulated in cancer over the period of time, how it relates to the change in the structure is the abrupt cell mass that has grown inside an organ. So this whole journey is we have to dissect that which one is the better call. So there are medical imaging where some imaging like CT, MR, what it really does for you is looking at the anatomy and the change in the anatomy. So essentially that means it is the structural change that through these modalities you are actually trying to look at. Versus there are functional imaging tools like positron emission tomography is the greatest, I would say, the invention to boost this field. And it's primarily the nuclear medicine technologies, optical imaging. So there are a lot of these toolbox that are already in place, which actually look at the change in the cell function, not the structure. So for say the cell proliferation. So there your requirement is or a cell death, like what Daira is studying the cell death mechanism in a viral infection case. So these cell death or even its movement ability, the cell's migration ability. So these are all the different functional property that has changed, and it can we probe these and measure these in the body. So those are the early events than a structural change that has happened in the later stage. Basically, what we need is a see-through procedure, and PETs, single photon emission tomography, SPECT, or even photon-based photonic imaging, which are optical. So they actually can help us in seeing through the body. So the body makes the body transparent, and look at where the different foci has formed, um, uh, other than the primary tumor, and all that thing. So we get a comprehensive look through this in a whole body scan. So as I've not drawn these basic background, so let me also complete it with this imaging. Now, imaging, in the sense, a lot of us actually who are biologists, we are dealing directly with the cell systems, and there is a huge growth of cell imaging and the development of microscope, starting from Galileo's identification of the simple compound microscope, where and then the coin of the first concept of cell coin, um, around 200 300 years, almost 250 years ago. So now, from there to the transmission and the scanning electron microscope, which came later in the around 1930s, between 30s and 50s. So these empowered us to see the complexity of the cell inside the cell, and the subcellular organelles, and how their structures are, how they look, and later on, it also helped us to understand their functions. And then in the modern biology is driven by the confocal microscopy, and there are many other uh, advanced microscopy that came in place, which actually helps us to see cells in a live condition, and different molecules that you are trying to monitor in a live condition as the cell go. So this journey is exciting, very exciting, and very important contribution it brings to the biological understanding for sure. But as I was mentioning, the body imaging is what the medical field is looking at, and there where the organ of the body scale imaging, and its journey is about 100 years, right? Um, so it started with that, the radiological diagnosis, where the first X-ray beam, which passed through the human body, and let us know that yes, there is inside the body there is skeleton. So there are bone structures which the X-ray cannot penetrate, versus X-ray can penetrate through the soft tissues, something like muscles and other stuff. Right? So from there, the journey started, and you see the nuclear medicine started developing since 1950s, between 1950s and 70s, and the ultrasound developed, and computed tomography, which is a modern form of X-ray, um, using computation, high-end computation, then magnetic resonance since 1970s onward. And the PET is the biggest invention that happened, and that bring out this whole field of molecular imaging as a that this is a completely different way of looking at the disease biology, where the focus is on the molecular functions, not the structures. So, and this whole game changer, this is my one of the favorite story. So there are a lot of important inventions that made these possible where we stand today in terms of imaging. But this story is always very intriguing to me. The story of Charles von Hevesi, right? So Hevesi is a radiation related scientist, and is a physicist basically, and his Nobel Prize won in 1943 in the chemistry. So his crazy ness has made his signature always. So in the Nazi time, he actually, in the fear that his novel would be taken away by them, so he actually dissolved it in acid and later on precipitated and reprinted to save that gold. This story is about he had possession of radioactive iodine, and this cartoon suggests that he is a paying guest and he had a suspicion that the landlady brings the old food back on his plate. So he sprinkled radioiodine after on the leftover food, and the following day he was scanned, and then to ensure that no old food is coming back, recycled back on his plate. And so this crazy journey actually says the power of nuclear uh, uh, elements, which are called tracers, and these use of these to travel over time to see the information um, uh, at different points of time. So as this is very simple from your physics concept that you all know, that energy and the wavelength and how their relativity is. So the shorter the wavelength, higher the energy, and the longer the wavelength, the lesser the energy. And we have elements like these: the gamma rays, the X-rays, ultraviolets, infrared, radio waves, and so on. And the only thing that we perceive is the visible light, which is a very narrow wavelength range between ultraviolet and the infrared, right? And this forms the basis of the photonic imaging that is covering bioluminescence and fluorescence. And the gamma rays and all, they are PET and SPECT. X-ray, it is a CT, NIRF and Raman spectra, which is actually the infrared area, and then the MR and the ultrasounds and so on. So there are in the radio waves. So just to touch upon, I'm not going to take deeper inside in the positronization because that itself is a topic. But just to draw your attention that yes, in the nuclear medicine, there are gamma emitters, beta emitters, and there are also positron emitters. So positrons are slightly different. They are positively charged, just like protons are, but they are not exactly protons. They are positrons. So when they collide with an electron, they release opposite angle, and this becomes the rays, which can be captured, and this gives the advantage of having a coordinate because it's nearly the annihilation photon is event is happening in the midpoint, and then you sense this ray and this ray. And because it is not a unidirectional, it is bidirectional, so you get a coordinate where exactly that event is. So this gives the positron emission tomography as a better imaging tool. And there are specific radio tracers which emits these kind of positrons, and that become the tool for this. And this is where this field required the knowledge of chemistry, intermixing the subjects, and to develop a probe. So, like for a biologist who actually work on the phase of identifying the target and perhaps also find a carrier. So these carriers can be actually turned into a probe called molecular probe to see. So now you can develop this construct with the help of chemistry knowledge, where you can, based on the modality that is available to you, you can choose a suitable level, like a nuclear level or fluorescent dye or something else. And then you can link it to the carrier. And then the carrier, if it is specific, sufficiently specific enough for the targets, so then it's a nice probe to see where this molecule is, what it does at different point of time. So this is how the concept developed. And there are, just to exemplify, there are several such positron emission and single proton emission type of probes that has been developed for different cancer-related functions. So something like cancer tumor metabolism. So there are radio-labeled glucose, amino acids, choline. There are tumor proliferation markers probes that are available, so something like fluoro, FLT, FMAO, and so on. And then the apoptosis related annexin V caspase related probes are already there in place, and tumor hypoxia. So cancer, as it grows bigger, so there are shortage of oxygen that happens, and that shortage of oxygen is causing the hypoxia. It causes the change in the pH, and all that parameter aggravates the cell's property, and that is also can be probed with something like F-MISO, ATSM, PASA, and so on. And there are also tumor angiogenesis. So as the cells become hypoxic, they start they are in dire need of blood supply, and so they start release the angiogenic signal so that new blood vessels can form towards to supply them. And these, there are probes, something like RGD, alpha V beta 3 related probes that is already in use. So, bringing back now, I would like to focus more on what this optical imaging world can do. And this is where this area is also for biologists. It's a great toolbox, and it's a very powerful toolbox with a diversified um, things that are available to you. And with this, so here you see that there are a lot of probes, which are primarily the dye, fluorescent dye, which when you excite with a certain wavelength of light, you get a different wavelength of light, which is of uh, shorter, which are of longer wavelength, so less energy. So if you heat with a blue light, you get a green light. You heat with a green light, you get red lights, and so on. And there are different forms of these. So there are natural dyes, fluorescent dyes, there are quantum dots, completely synthetic, there are carbon dots. So many forms of these are available in the material world. And there are also probes that support something like from the biology. In the nature, they naturally persist. Just as a mating call or catching prey, these there are different zoonotic elements, and the components have been identified, and now through recombinant DNA technology, they are being applied, and cells can be engineered for these cells that has been harvested from non-mammalian sources and utilized when they are basically enzyme substrate reaction, and that glows in the dark. So this is very attractive because this naturally reduces the background signal and bring a specific signal at the site where the luciferase is present and it is finding its substrate to catabolize and produce the light. Versus here, you have to shine a light, so you are already blurring the area. And then if the signal is very weak, you may have difficulty to see the contrast that develops. So that's the basic difference. But over and all, both the tools are very useful, and they have. I'll just exemplify a couple of things for the sake of time, very quickly. That these, just for say the bioluminescence imaging, how it helps us to stitch. Conducting experiments at multiple different scales. So starting from your simple recombinant DNA clone picking. So where bacterial colonies that has come up, you want to identify which are the one that are positive, and you spray the substrate over the plate, and the enzyme is already present in the colonies. They will glow, and like this. So it readily help you identify which are the clones that are positive. If you are advanced from there, and then you have that plasmid you have expressed in a mammalian cell, and you are trying to identify the clone population of the mammalian cells for your further processes. So you can also identify them in a plate. So the cells are in live condition in the media. You just add the substrate in the media, and they will start glowing, and you identify which are the ones that are glowing. And then in a model system, so there are different level of models that people use. Some use plant models, other vertebrate models like geographies, or even mouse models. So these are the preclinical different models that people use. And you can have your genetic sensors integrate in them, make transgenics, or make engineered cells like human cancer cells engineered and then delivered, and see the kinetics of the tumor growth and its metastasis, and identify clone to clone difference in cases over a period of time. And this all can be done in a non-invasive manner because these substrates are not interfering the body's thing, and they are not toxic. And the enzyme do not change the cancer properties per se. So it is a great tool to see the things of the event, the chain of events that happen over a period of time, right? So just another example from my own group's work, like another way, like we implanted a few human cancer cells in the mouse memory pad. And as I mentioned, we deal with breast cancer. So it's the mammary fat pad. We place the human cancer cells, which are leveled with the luciferase. And within a period of 2 weeks, you can see the tumor has grown, and then it starts spreading. And in a control case and in a drug resistant case. So these cells are a lot more aggressive, and within that same period of time, you can see how the difference is in terms of control. And then at the end point also, you can dissect out the ex vivo confirmation can also be done, that where the tumor cells have spreaded, it has spreaded to bone, it has spreaded to brain, lung, and liver. Uh, I also talked to you about the use of dyes. So this is one great example from our own work. Is we synthesized a novel dye which has a fluorescent property, and then it also can trigger heat when it is triggered with a photon beam. So at certain wavelength, this dye captures the beam, and then it resonates. And then that resonation actually causes the heating. And because of the heat, the cancer cells can die. So this is what the you can actually do. And when such fluorescent dyes are very important because they support the theranostic purpose, because you can utilize the fluorescent property of it for checking its distribution over a period of time, and then its accumulation in the tumor. And then once it is accumulated, you can trigger it with the laser, and then it causes the heat, and then the tumor get charred, and then the tumor get healed. So this is called photothermal therapy, which is a few students in my group are also working on this aspect as a future cancer therapy. The other thing that also comes in place in optical edema is also very important for radiochemists who deal with a lot of radiation and make probes for molecular imaging purposes. But their exposure to radiation is always a threat. And right, so they get actually burnt out by themselves because they are dealing it with their hands and everything. So the radiation exposure itself is a threatening thing for them. So for them, now the positron or SPECT scan are a little bit longer, and it increases the exposure time. So what they do is now this Serenkov luminescence. So when a radioactive, like a positron, it travels in a dielectric media in the speed of light. So they emit luminescent photon, and this is the event that was identified by this scientist, Russian scientist called Serenkov. And this is very intrigued because this is as per the quantity of the radioactive. So this is a serial dilution made, and you see the photonic signal is actually varying as per the strength of the radioactive amount present. And how do we know that it is really optical or photonic in nature, not radioactive? If you cover with just a piece of black paper, so the same amount on the upper and the lower side, you can see the top part we have covered with the black paper, and you cannot see any signal. So no photon can pass out this black paper, right? So, and it is very good. So it is a very nice tool using, and it's a much quicker scan that you can perform. So we actually use this for distribution of iodine 131 in thyroid. And so here this mouse received the iodine 131, and it is accumulating in its thyroid. And this is the bladder and the tumor where it is also accumulating. So this is just to exemplify, and there are many such probes which have Cherenkov signatures. Some are beta emitters, some are gamma beta emitters, and but they all have the Cherenkov signatures which can be utilized. So it is one probe that can cover two dimensions, both the PET or the SPECT scan based on the particular tracer that you are using, and then you can also perform much quicker optical scans using these. The system, I had the privilege of witnessing this whole journey of the invention of this black box CCD technology for optical whole body imaging for preclinical experiments. And this is one of the beginning equipments where it used to, in the early days, it used to be only the planar image, meaning it is just one plane. You don't get reference of the body depth where the source is located. But now today that system has upgraded to a system called 3D system, and where you get a completely tomographic image that can be generated based on the bioluminescence or the NIRF fluorescence. And you get to see in respect to the rest of the body's three-dimensional aspect, the depth, the volume of the tumor actually. So all that can be measured. The other way of taking the molecular function to address the molecular function. So these are, so far what I talked is imaging how it is propagating momentum in terms of our visualization ability over a period of time in a non-invasive manner. So now you can utilize those things in terms of therapy monitoring, disease monitoring, recurrence, and all that aspect. There are also molecular aspects. Now, if we now focus what you could do in the cell, can we do it using in vivo? So from thinking from that perspective, we started looking on developing the molecular sensors. Now, there are biophysical principles, so something like Jablonski diagram, if you per se. So if there are compatible elements, moieties are there, and which have an overlap on their the emission is overlapped with the absorbance of the second moiety, then there is a transfer of resonance energy. And that resonance energy can excite this. So you are not actually signing the light that is required for this moiety, but you are getting its output because it is getting to an excited state by absorbing the energy. So what we see here is that, so using this is called a BRAIT imaging assay. Here the donor is a luciferase and the acceptor is a fluorescent. And we can, in the linker, we can place two interactor proteins. And as they interact, the proximity, it's a very proximity-driven approach. and it's only works in less than 10 nanometer physical distance. So if this is a tight interactor, then these two moiety comes in the proximity, resonance energy transfer happens, and then you get the affected signal just by adding the substrate for the luciferase tumor. So, and these are the different principles. And again, just like freight, a lot of you might have used these or will be using these freight fluorescence resonance energy transfer. Here, there are two fluorescent moieties involved, and here the donor is a luminescence, and the fluorescent moiety is the um, acceptor molecule. And you can have the same similar setup, the donor channel and the acceptor channel, and measure the outputs. And then ratiometrically, you can actually, uh, uh, based on the ratio, you can determine what is the distance and what is the interaction strength and all that. And how, if there is a disruption of that strength, how that is affecting that strength. So this could be a nice assay for screening drugs and so on. We we have worked on this in the past. I'm not exemplifying. I'm just trying to focus on the latest that we have done in my lab here. Is something like we challenge this dogma of protein phosphorylation, which is the standard assay that we measure the how when a protein get activated by a kinase action. This how, this is a very fast process, and this is the basis of a lot of cellular function it controls. And there are about 30% protein which in the human genome that undergoes the phosphorylation. So this is a very, very important event in terms of cellular signaling, network understanding, and all that. So here we took this challenge to monitor STAT3 phosphorylation action. And what we see that in the STAT biology, what we see is when it forms a homodimer by getting recruited in the JAK kinase. So it forms a dimer, and then it translocates to the nucleus, and then it becomes a transcription factor for multiple genes which are oncogene related, oncogenic function related. And but what we see that there are many two major two major sites where the phosphorylation happens. One is the tyrosine and one is the serine at very close by positions of 705 and 727. And but there are finer things in the cancer. Now, where most of us, until we studied these, so most of us, we always focused on canonical activation, that is Y705, tyrosine activation. What we see in TNBC, it's a type of triple negative breast cancer, which is a type where there is no targetable drugs that are till death, because there are no receptor that can be targeted. But there are other mechanisms that people are started targeting now. But in this type of tumor, what we see is that when a STAT is present, but it's not, it's through its kinetic, it's not, the STAT function is through the 7 to 7 serine phosphorylation, not the tyrosine phosphorylation. So it perfectly correlates. The scoring is mostly in the same tumor sample. You see the STAT is present, it is the activation of the STAT 7 to 7 is present, not 705. So it is operating through a non-canonical axis. And we actually aimed this to develop the biosensor so that we can distinguish this and we can screen drug that can help to nullify this action. So we developed this great approach, and by bringing the stitching through recombinant DNA technology, stitched STAT3 whole protein to the nano-look, which is a luciferase, another type of luciferase from a marine organism. And this is the acceptor molecule, turbo-FP, which is 635. It's one of the farthest fluorescent probe that is known, fluorescent protein that is known. So they are genetic reporters. Now, by co-expressing these four constructs in combination, you can see a great signal is in the presence of EGF, which is the trigger for the homodimer formation of STAT3. It is forming the homodimer, and the red signal is increasing only by when we co-express NLUC and turbo. So these two, when they are present, and you add the trigger, which is the activator EGF or IL-6, so you see this kind of change. It separates out from the baseline. So we use this for screening a lot of different drugs, arbitrary drugs, along with the control. So something like EGF is the trigger, and then there is a known drug for that inhibits the Y705, that is static. So this definitely inhibits the action of the STAT in triple negative breast cancer cells. But we then found that there are multiple others which actually inhibit much more intensely than static. And this, so niclosamide, for example, it's an anti-helminth drug, and it can be repurposed, which could act as a much better inhibitor. There are many other candidates that we are now working on. And just to finish this part with an example of what niclosamide do as compared to static, you see that static can specifically in the presence of static, the specific Y705 band get abolished, but it has no action on the 727. But when you add the niclosamide, you can see that both the Y705 and 727 goes away. So, and it affects the cell's survival ability. So the number of colonies that can survive and form colonies, so they are minimized when the cells are treated with the niclosamide. Another aspect, for the sake of time, I can skip this up. So this is just to bring more confirmation on a different biology aspect. So this is another, but if it is required, I can stop here and I can summarize, and I start taking questions and interact and better. Or if you allow me a couple of minutes, I can just quickly get it through.
Yes, sure.
Okay. This is another type of breast cancer where HER2 is an oncogene, and it's a receptor tyrosine kinase. And you see that HER2, and there are multiple other members. So HER2, HER3, HER4, and EGFR, which is HER1. So there are, it interacts, it naturally forms a homodimer. And there are specific drugs like trastuzumab or Herceptin, which is from Genentech. So these, and there are many other forms of drug that actually inhibit this HER2 homodimer. And this has worked as a personalized drug. And the last 15, 20 years, they have been in use in the clinic, and they're of great savior. And this subtype of breast cancer is actually well controlled when they see this drug treated with this drug. But while the treatment, it is almost invariably any drug that you use, the cancer cell will bypass that at some point of time. And so has happened with these even costly drugs, and the tumor relapse in a later phase of time within a couple of years or so. So 5-year survival has definitely showed improvement, but there are inevitable bouncing back. And why it bounced back? And this is where we started asking questions. And so one of my PhD student who have finished and now started a postdoc in University of Lausanne is on the interaction domain of HER2. So the interaction phase, there are a couple of hotspot mutations that we identified, and some of them shows the deleterious through the different bioinformatic tool. And when these deleterious mutations over the unaltered group we compare, we do see a survival difference from the TCGA data. So we generated clonal establishment. We made the clones with the mutations in the HED2, and then we express them, overexpress them in the clonal cells. And when we treated them with the trastuzumab of different concentration, 100 and 200 microgram, you can see for each case. So the wild type and the 305, one of the mutations, they continue to respond to trastuzumab, but a couple of other mutations like 309A, 310F, 310Y, and 523S, they are less responder or no responder. So this brings a very this was kind of unknown to the field, and we actually investigated. We spent the whole 5-6 years now in understanding the whole underlying biology, what it causes. So what it causes is actually it causes the when head-to-head to homodimer happens, it is operating, the downstream signaling is operating through ARC. But when these mutations cause it, make it more prone for heterodimer formation with head3. And head3 is a pseudokinase, and so now the head2 is actually operating through head3, and then it is triggering the PI3K AKT kinase in the downstream. And this is the reflection in the Western blot. And so we had these other phosphosensors made for AKT and ARC phosphosensors for a different project. And we thought then now we are seeing, because of the homo or the heterodimer, we are seeing a signaling switch happening, right? So we generated the engineered cells with these two neo and nat phosphosensors inside the cell, and and we actually measured for each of these mutation lines what is the change. And we do see that yes, it is the ARC sensing that sends higher signal in case of the wild type of the 305. But for the other three new mutations that we found, 309, 310F, and 523S, so they are more AKT oriented. And this is for the ARC sensor, and this data is for the AKT sensor. And so here you see the wild type of the 305, it is not operating through the AKT, but it is more operating through this. So this is the application of BRAIT in a much complex biology. So I'm not taking you deep inside, just to exemplify, I use this as a model that how we can really sense the molecules in action inside the cell, and how optical imaging can help us to visualize these actions. And then we can actually use, once we know, then these assays also help us to evaluate the inhibitor molecules or any other way of controlling mechanisms, so that this molecular event can be stopped early on, right? So with that, this is the kind of summary I would say that I know I prepared this talk for more basic, so that you can connect better to this complex world of imaging where multi-field is contributing. But these molecular, I've tried to impress you upon that the dependence on functional imaging is very important over the anatomical imaging, because that is a change that is much later change. So if we really have to address cancer in an early stage, we must focus on the functional change. And if it comes through the imaging, then you are empowered to do your process and run your screens and everything in a much early stage change that you can bring. Both radioactive and photonic based probes can be made, and I have shown you how the differences between tracer to probe. The probe is actually integration of the tracer along with a biological moiety through a chemical linking process. And there are also genetic based reporters systems like luciferase or frozen proteins that can also be used, and they can also use in a combination to form molecular biosensors, which are uh, very much doable within our own set of knowledge of recombinant DNA technology. And then some of these specific biological context, like stat function or the head-to function, how these molecular applications can come in place. So that's all I have. I hope I have left a lot of time for you to interact, and I'm happy to take further questions that you have. And just before I stop, the last sentence is this is my group who keep me alive and excited all the time with all that effort that we make. And some of the examples that I have taken are contributed by the students who have mostly finished, and they have moved on for their next career move as postdocs in different places in the world. And I have collaborators and funders. And I thank you. I'm happy to take questions now.
Thank you so much, sir. It was really insightful talk today on imagining techniques in vivo and also in vitro. So I would like to start the conversation by asking you one question. Not much in vivo, but since I work more in
Excuse me,
vitro,
I'm
so
so sorry
like.
to interrupt, but your
Uh,
voice is echoing for
now,
me.
now it's better. Now it's better. Yeah, yours is better, Bhakti. But Daida's is it was like I was trying to make up, but yeah,
Uh-huh.
Daira, go on. I will make it up.
Can you hear me, sir?
I hear you. It is like my screen. There is a cartoon happening in
Okay.
your voice.
Okay. So I wanted to ask you that since you worked on STAT3 activation and its phosphorylation, did you also try different inhibitors and try inhibiting the STAT3 activity and see that like which phosphorylation sites, like you showed you niclosulfonamide, I guess. Did you
Niclosamide.
also try?
Uh,
Did you also try any other inhibitors?
Yes. So the STAT3 is a transcription factor. So there is not
Mm-hmm.
much hope that you can tamper with the STAT3.
Yeah,
So
yeah,
even though it is because it controls it's like P53. It's a
yeah.
known thing, but we cannot really tamper much because it also
Mm-hmm.
affects the normal function. So
Mm-hmm.
STAT also has that. In that sense, STAT do not have too many inhibitors around, but there
Mm-hmm.
is the specific inhibitors. Of course, there are there, and static
Mm-hmm.
is the one that we have tried. And static, the
Mm-hmm.
reason we tried because the normal function it stops the canonical. It's a
Yeah.
inhibitor for canonical function that tyrosine
Mm-hmm.
kinase. Um,
Mm-hmm.
so uh, so we use that in our screen, and we compared with multiple others.
Mm-hmm. Mm-hmm. That's really nice. So we have few questions from audience as well. And one of the question is on okay. So uh Tamogna Chatterjee, she wants to ask that on basis of what parameters the cellular targets of the cells are identified for imaging?
Okay. So you know, very good question. Very good question. I wish I would have touched upon on this. Uh,
Mm-hmm.
so the I by asking this question, I mean, I think her intention is to ask whether the uh, target is present on the membrane or intercellular.
Mm-hmm.
So yes, so because it depends, right? So, for example, if you are using glucose, then the glucose is getting phosphorylated inside the cell, and so it will get inside the cell, and it will get converted and then get trapped and accumulated. So now if like FDG, which is fluorine
Mhm.
18 leveled glucose, or a fluorescent tag glucose, so they both are present in the current time. So you can use these, and this is an example of cellular uptake of
Mhm.
the glucose in the form, and then it get trapped, it is accumulated, and that accumulated signal you are trying to sense. There are also receptor based, like RGD peptide. So when it is a peptide or an antibody based probing that you are thinking about, then
Mhm.
perhaps it's important that you target a receptor or a protein that is present on the membrane, so that it it do not suffer from internalization issue if there is any, and the bulk could be the moiety's bulk could be a issue for that. So these are the factors that one has to keep in the mind, that what can really be converted into a probe and what should be a targetable probe for that. Yes,
Mhm.
that's a good question. I hope I
Great.
answered this. Yeah.
I hope that sir answered the question. So there is a question from Alka. So she finds molecular imaging very interesting, especially in vivo. So, like, what are the biggest challenges in the field currently, as per your experience, that needs to be addressed? Because I have also heard here from people who do molecular imaging that sometimes for the mouse, they have this transparent window kind of thing stuck. Let's say somebody wants to see the gut, so they have something like transparent um,
Yeah,
sheet on, and then
it's
they
primarily it's called, yeah, I believe it's a Rakesh Jain's group actually who work on this angiogenic
uh-huh.
factor. So they use this a lot. It's called window chamber model.
Mm-hmm,
So
mm-hmm,
it's a mouse screen. Screen is means stretched and clipped with a glass window, and this is
mm-hmm,
where this is required when it is a something like intervital microscopy,
mm-hmm,
which you can actually trying to see the molecular events that is happening in a live animal,
mm-hmm,
but it is like the vasculogenesis aspects of new vessel growth and all that, in the live mouse, and under a microscopic objective. So
mm-hmm,
there the issue is because the breathing, like we breathe, so the movement of the body that itself is a distractor. So
mm-hmm,
engineers have to address this breathing movement. So how
mm-hmm,
you nullify these, so that, because to and when it comes to under an objective, it's a huge movement you can imagine. All of you are using microscope, so
mm-hmm,
perhaps you can imagine this better, that how a movement of few millimeter, what it makes
mm-hmm,
when it comes to watching things under an objective. So
mm-hmm,
that's where the intervital microscope, where it is through the objective, you need to by placing the window chamber, you are actually stiffening that area so that it is out
mm-hmm.
of the breathing movement, and then it you can resolve what you were trying to resolve.
Great, great, sir. So, Somalia wants to ask a question that how you currently in the imagining technique, how is AI helping us, to
It's
improve the quality, or whether is it like how is it going to get in future?
it's huge, 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 because it is the body imaging has become a big guidance for clinic, and
Mm-hmm.
the decision making of what should be used, and also after the biopsy, the confirmation from the radiology is also a very, very important component. And all these radiological parameters, so they are all integrated with machine learning and AI. And,
Mm-hmm.
and the anatomical imaging, I was telling all of you all this time now that anatomical imaging has constraint. Um, I was raising this point particularly because the anatomical imaging is the image that you generate until and unless a sufficiently resolvable structural change happens, it would be
Mm-hmm.
read as a normal. But basically there is something that has changed, but it was not within the ability of the imaging system's resolving power, and that's
Mm-hmm.
why it is like a negative, and so it's normal, but there is a change. So that's where I was drawing this, and AI actually is pushing this boundary, because this machine learning and then application of AI brings further confirmation to nullify these false negatives or false positives. So increasing the specificity and the accuracy of prediction of disease. So it
Mm-hmm.
has a huge impact, and it is already impacting. It is already in
Mm-hmm.
place, and it's a great tool of uh, yeah.
Mm-hmm. Great, great. Sir, so uh, I think, yeah. So here, yeah. So Abhida is asking a question. So it's a bit targeted to myeloma cancer, but I would rather, take it also a little bit in general. So the
Yeah.
question goes like that. Uh, can I would simplify it and say that, can you detect, early cancer through this imaging technology?
Um,
Or
I
how is it done currently?
Yeah, of course. There are, these some of these probes that have already pushed the boundary. Um,
Mm-hmm.
the there are probes for there are probes that are already in place that are early cancer target that people have identified, and um, of course uh, there are adaptation of let's say liquid biopsy is one thing, or a blood-based biomarker, or a PCR test that has been performed before, like prostate-specific antigen or CEA. So these are the kind of elements. So there are prostate-specific probes that has been made, and so instead of running it from a blood test, you can also do that using that specific probe wherever it is available. It is availability is not widespread all across the world. That disparity is still there. So taking that admission, but it is not that the probe is not there. It is the probe is
Mm-hmm.
there. And they are definitely, like City of Hope is a great place. So where a lot of antibody engineering happens, and I know Professor Anna Wu, who is the president of World Molecular Imaging Society. So her own research is about these antibody engineering and converting them into probes for PET probe or SPEC probe. And her focus is on these kind of things, that the antibody based detection, it would be very specific, but then there are a lot of disadvantage because of its bulk that it had, and then the probe's distribution it affects. So she is engineering these antibodies as single chain antibody or scfb and so on, and reducing the bulk, and then it is actually keeping the specificity. There is also another startup company that I know of is in Sweden, is Affibody. So they are bringing out a peptide like a moiety called Affibody. And these
Mhm.
Affibodies for EGFR, HER2, they are already converted into probes, and they are for detection of HER2 positive or even EGFR is for lung cancer. So there are body probes that are available which detects early on for the biomolecular uh, function that is already well established in the clinical literature, that lung cancer for EGFR, then this antibody for EGFR can be used for a diagnostic probe. And so we see the accumulation overexpression of that in an early stage. So yes, it is possible. The disparity of distribution is an issue that has to be admitted. Yes.
Great. Sir, so Dharmesh is asking a question in this regard only, I guess. I had that what are the contributions of the nanobodies and the nanorobots that are coming up today, and how are they aiding us in the microscopic techniques? And can you also engineer them in vivo imagining, especially?
Uh, why not? Um, Yes, nanobots are very, very interesting objects. Of course, you have to orient them through the magnetic field, at least for some of the bots that I know of. With the magnetic field, you actually orient them through the cell and then take it where you want to take them. Now, if this bot is labeled, then this that signal. So one has to devise an integration of the imaging tool along with the orientation tool for the bot. If there is a bot nanobot that is self-operable, then perhaps that integration is not required. But nothing of that sort I am aware of as of now. But
Mhm.
yeah, I guess they are very exciting tools that are in generation, and in future run, I'm sure. I mean, for nanowatts can be used for delivering materials as well as they can be used for tracking through
Mhm.
imaging.
Mhm.
Yeah.
Mhm. I see. Sir, so there is one more very interesting question, so I would like to
Yeah,
take up.
most questions are very interesting actually. So it's good.
So Rika is asking that what are the advancements for in vivo imagining that can help in protein-protein interaction involving phosphodiesterase in cancer?
Phosphodiesterase as such, I don't know, but one can think of
Mm-hmm,
exactly what can be done. Um,
mm-hmm,
the protein-protein interaction, we have many tools. My group primarily we have worked focused on utilizing the BRAIT assessment that I explained.
mm-hmm,
But there are many forms of these that can be utilized for identifying protein interactions. So something like reported complementation.
mm-hmm,
Um, you can chop a reporter protein into two pieces. So there is no individual component do not have any reporter activity. But if through the interaction, if they are complemented, then you get that complemented reporter activity. So a gain of signal that happens through the interaction. So this could
mm-hmm,
also be an approach, and it is a viable approach. Plenty of literature you can see. Luciferases are splitted into two pieces. GFPs are splitted into two pieces and utilized. There are also combination of these. So something like biopsy, and there are many such strategy that is. So where the like the donor, like now this is one to one interaction is one simplest thing. The in nature inside the cell there are even complex interactions, where one protein then it interacts with another protein, and then it can bind to a third protein or a fourth protein. So the requirement is much bigger than that, but simplest form is this one to one. And thinking all that, there are a lot of variables that has been developed as well,
Mhm.
variable strategies. So something like intermixing of split reporter along with rate or freight. So each of these donor are splitted, and they are through the interactors, they are coming, and as the whole complex is forming, then the transfer of energy will happen. So uh, but yeah, what you can the tools, good part is the tools are very sensitive today. So the optical toolbox that I showed, they can in fact detect sufficient signal, photon signal from single cells. So the tools of the CCD camera, the nature of the camera that is available on these and mounted on these systems, they are very exquisite power they have for detecting photons. So that way, I think it's a very exciting field. Phosphodiesterase, for say, um, I haven't gained much knowledge on that part. So, but I uh, either one has to try that part. I don't know. I am not aware. So, yeah. But application wise, the assessments are available. One can
Mhm.
design and sensor and do it if not done.
Mhm. Great, great. So I would like to take like two last questions.
Okay,
One is from the audience and one from my side, which I asked to almost all the PIs.
fine.
So the question goes like that: are there any specific imaging modalities best suited for studying cyclic nucleotide signaling disruption in cancer?
Yes, it is there. There are
Mm-hmm,
there are braid probes, there are fed probes, there are synthetic
mm-hmm,
probes that are there.
mm-hmm,
And these braid
mm-hmm,
plate that I have mentioned is primarily the genetic probing,
mm-hmm,
meaning it's the recombinant DNA, and then you express them, and then they start sensing the cellular change of the function. But there are also probes, like it could be semi-synthetic, like
mm-hmm,
donor is a reporter, and then you use the activator, the acceptor is a probe, which is synthetic in nature, chemically synthetic, along with a fluorescent dye or something like
mm-hmm,
that. So we have also demonstrated such strategy recently with by working with a group in ISA Pune here in chemistry group. So for caspase activation, yeah,
mm-hmm,
caspase 3 activation. So we have ACS pharmacology and translational science. We have a paper last year. So we actually use this JHAD FMK, and it is fluorescently tagged, and that is a ready available probe
mm-hmm,
from the company. And
mm-hmm,
we express the caspase luciferase construct inside the cell. Now,
mm-hmm,
as we are adding this, so this interaction is um, now if you want to screen the apoptosis inhibitor that is preventing this caspase activation function, so that can be detected using these probes. So yeah, so these are called semi-synthetic braid or freight. So one side is genetic, so it's less complex, so that only one plasmid you have to express and select, and then that selected cell line you can use for screening by adding the probe for the acceptor part and along with the inhibitors and all that complex.
mm-hmm,
So
mm-hmm.
yeah, and for amp cyclic amp, there are great probes I know for sure. Yeah.
Great. So since I work on caspase 3 and apoptosis, that's why that's a
I'll
very
I'll
relevant
forward you.
model.
I'll forward you my paper. You take a look.
Definitely,
Yeah.
sir. I would really like to read and the structural moieties of caspase 3. Great. So we would wrap up this event. So I would like to ask you the one last question, which
Okay.
I usually ask to all the scientists which come here.
Okay.
So what is your favorite discovery in the science so far, apart from a microscope?
All right. Yeah, I think this great invention,
Mm-hmm.
um, it has been very exciting to me.
Mm-hmm.
Um, my PhD was in entomology. Okay, so
Mm-hmm.
from the insect world, I come to now human world of cancer. So it's a
Mm-hmm.
long journey, but you know, human nature is such that I don't want to miss out my links, past links, even though they are
Mm-hmm.
past. But we find a match when the past matches with the present, right? So,
Mm-hmm.
so when I got a hand first time in the imaging world, and
Mm-hmm.
this toolbox of optical imaging toolbox that was actually born in front of my eye. The first company was Genogen in Bay Area. So they invented this box first time, and when that box came, and we get to see that a firefly luciferase, uh, how it matters for visualizing molecules, then it was a exciting match for me. And since then, I took a bigger challenge that can we take this simple imaging strategy to more complex understanding, where biochemistry have always been challenged. That running a co-IP experiment is always the gold standard. So can we get in that dogma, and can we see
Mm-hmm.
uh, proteins interacting in as it is inside a live
Sure.
cell? And if
Mm-hmm.
that was the development that I have made, that has grown through my hand, and it's definitely one of the most exciting thing that I've done in my life. So yes, but I know the modern day, it gives a lot of opportunity to do many good things, and we are doing some of that to the extent possible. But yes, I think the science field as such is in a state that is really really very exciting, and this is one touching story that I'll definitely bear in my mind for some time.
Great, sir. Thank you so much,
And
sir.
very
Sir,
good
so we
introduction
would
also.
like to, sure. Sir, so we would like to have one more session round with you next time whenever we get time and you have some available slots, and we would
Always,
like to
always.
delve more on your journey and on the imagining and how you surpassed various challenges in the experiments and things like that. That would be a really motivating session for the students as well.
Excellent. Yeah, I'm. It's a lot of good questions has come through your session, and I really liked all of your motivation for gathering together in this format, whichever way you have devised it. It's a great initiative, and always feel free to reach out to me if I can contribute in any way and help you on your initiative this way. So
Sure, sir.
it's a great pleasure to talk to all of you, and thank you, thank
Sure,
you.
sir. Sir, so we also have Janvi here now. I
Yes,
guess can we
I
hear
see
her?
her. I see her screen. So there are now six. Samlia, Bhakti. uh,
So most of our people are on YouTube live. Like we had around 110 on plus registrations. So people were really excited to listen to you today. And honestly, it was a really great and motivating talk that what we can do by imagining today in the in vivo especially was uh, great. So now I would like to take ask Bhakti to please deliver vote of thanks for our sir and audience.
Thank
Okay,
you
thank
so
you,
much.
Nadia. Yeah.
It is my privilege to extend a heartfelt vote of thanks on behalf of all of us here. First and foremost, I would like to express my sincere gratitude to our esteemed speaker, Dr. Abhijit Dey, for sharing their valuable insights and expertise with us. Your engaging presentation has been both enlightening and thought provoking. It has left us with valuable takeaways and a fresh perspective. I would also like to thank all my teammates for their efforts in putting together this event so seamlessly. From coordinating the schedule to ensuring a smooth flow, your dedication behind the scenes has made this webinar a success. A special thanks goes to our audience. Your active participation, insightful questions, and enthusiasm have truly enriched the session. It is your engagement that drives the success of events like this. Once again, thank you all for being a part of this wonderful session. Let's continue to think, grow, and share together. Have a great day ahead. Thank you.
Thank you, Mukti. Thank you, Dhairya. And thank you,
Thank you so
Vaishnavi.
much.
Okay. Thank you, all of you. And wish you all the best in your journey in the career. And please reach out to me at any point of time if you need any help. Yeah, we'll be
Sure,
happy
sir.
to be part. Yeah, and good to see my
Definitely.
student Janvi is also part of your initiative. So, yes, Samila, Shamil, how do you pronounce?
Familiar.
Shamilia, Misra. Yeah. So it's really a pleasure to meet all of you here, and thank you so much. Thank you. Yeah.
Thank you so much,
Enjoy your
sir.
rest of the Sunday. If you are not
Same
working.
to you, sir.
Thank you. Bye.
So, guys, feedback form will be posted in the YouTube live chat. You have to fill it and then you'll get your e certificates.
Absolutely, your feedback is very important.